Hello students, a very good afternoon. So today we'll continue from where we left off from yesterday's class. That is, we have to discuss from immune system. So before we start today's session, so I would like to launch a technical poll. Please let me know if the audio and the video is clear. Yes, the technical polls are live now. A quick response would be helpful. Students from YouTube, if there is any technical problems, you can just type your issues in the chat box. We'll look into it. Okay, students, thank you. So I will end the poll. If in case you have any issues, be free to mention it in the chat box or raise your hands. Anything would be helpful. Okay. So let us start our today's session with a question. Fine, you have your first question, that is the letter T in the T lymphocytes refer to. Okay. Your options are thalamus, tonsils, thymus, and thyroid. So I will launch an MCQ poll. You will have 1 minute to answer this question. Make it as quick as possible because this was discussed already in the last session. Okay, your poll should be live. Yes. Come on, make it quick. The letter T in T lymphocytes refer to. YouTube students, be free to put it in the chat box. All right, so let's end the poll now. Your time is up. Okay, let's see what is the right answer. See, the T lymphocytes, they are formed, okay, or produced in the red bone marrow. So once they're produced there, they go for maturation to the thymus. Since they mature in thymus, they are called as thymus lymphocyte cells, or in short, they're called as T lymphocytes. So our answer is option C, that is thymus. It's a very direct question. So you can answer this as quickly as possible if you remember. Okay, next one. Which of the following acts as a physiological barrier to the entry of microorganisms in human body? Your options are epithelium of urinogenital tract, tears, monocytes, skin. So we have our options now. So let me launch you a poll. Okay, the poll should be live. Yes, you have a minute to answer this question. Let's make it quick, students. Okay. I would appreciate if you do it in within 30 seconds, Which of the following acts as a physiological barrier to the entry of microorganisms in human body? All right, so let's end the poll and see if your answers are right. So here we go. So when we let us see the question. So the question asks about which of the following acts as a physiological barrier. So let's take individual options and analyze them. So the first option, that is epithelium of urinogenital tract. So epithelium of urinogenital tract is an example for physical barrier. Next is option B, that is the tears, right? So the tears that are produced in the lacrimal glands, okay, they have the composition of tear is antimicrobial in nature, and the pH also is antimicrobial. So the tears is an example for physiological barrier. When it comes to monocytes, monocytes are an example for cellular barriers, and skin is also an example for physical barrier. So our option for this question is option B, that is tears, which is an example for physiological barrier. So we will just try to revise from where we left off yesterday. Yesterday, we were talking about immunity. So we divided the immunity into two types. One is innate immunity, and the other one is acquired immunity, correct? No? So now when we talk about non-specific immunity, so we have physical barriers. So we gave the examples of physical barriers, that is skin and mucous coating of the lining of respiratory, gastrointestinal, and urinogenital tracts to trap the microorganisms. So right. So under physiological barriers, so even they have the properties of preventing any of the antigen infections. Okay. So this happens at the physiological levels through cellular secretion such as gastric acid or the secretion of HCl in your stomach. Okay. And the saliva have lysozymic properties. The tears in your eyes. See, there are cellular secretions which prevents the microbial infection. So that is why we call it as physiological barriers. Okay. Now we will talk about cellular barriers. What are cellular barriers? See, there are some of the white blood cells which are protecting our body in a very non-specific fashion. They do not care about what type of the antigen has entered. They just care whether it is an antigen or not. Okay, whoever it is, irrespective, it might be a dust, it might be a bacteria, or it might be a protozoan. Okay, so they just understand that this is outsider and they will go and attack them very non-specifically. So what are the types of cells which participate in such non-specific immunity, such as neutrophil cells, which are also called as polymorphonuclear leukocyte cells or PMNL cells. Okay, we have monocytes and natural killer lymphocytes, as well as macrophages, okay, which are the cellular barriers or a type of WBC cells which participates in non-specific immunity. Okay. And next one is cytokine barriers. Cytokine barriers are a very interesting type of barriers that we can understand. See, let us assume that one of our cells, our own cells, are infected by virus. See, the problem for our body is whenever the virus is entering in our body, it is very hard for our body to recognize, okay, these viruses, because they have exceptional ways to cheat our or blind our immune system. Okay. Now, once the virus has made an entry, it infects the cells. So once the virus infects the cells and the cells, functionings are modified, then the cell will understand that it is under attack. So if I am under attack, okay, so would I warn my neighbors? Would I warn my friends and all those things and all the other important people around me? Definitely, right? Similarly, even our own cells, what they do is they warn the neighboring cells about this attack, okay, through a cytokine protein. Okay, they produce something called as cytokine protein. It's like a signal. These cytokine proteins are called as interferons. So once these interferons are produced, they go to the neighboring cells and they warn these neighboring cells and they make these neighboring cells capable of blocking a viral infection. How do they do this? So they make the, once they warn these cells, the cells which know that there is virus around them, they produce some of the proteins which can block viral replication. Okay, and they even block some of the bind. For example, there are certain proteins that help viruses to replicate within our cells. Okay, so they produce these antiviral proteins so that the infection is not happening in the neighboring cells. And that is possible by the signals that are produced by already infected cells. And these signals are called as interferons. And these type of barriers are called as cytokine barriers. Okay. All right. So let us talk about acquired immunity. What is acquired immunity? As I said, acquired immunity is a very, very specific type of immunity. It knows what it is fighting. It knows how to recognize, okay, a particular antigen. It knows the type of defense mechanism it has to launch, okay, to neutralize this particular infection, right? That means what? It has the capacity of remembering, right? That is memory, and it also has the capacity of launching a very specific attack to neutralize this particular antigen. Okay, so let us say an individual, if you yourself, okay, should have the capacity to recognize someone very specifically and interact in a very certain way. Okay, is it possible for you to do that in a very first meeting? Okay, if I meet you for the very first time, is it possible for me to recognize who you are, what is your name, and where you're from? It is not possible. So first I need to have an encounter. First I have to meet you first. Okay, the more times we meet, the more familiar we get, and more easy it is for me to recognize, remember, and easily communicate with you. That is the same mechanism that happens in our body, correct? No? So what is happening in acquired immunity? As I said, it is pathogen specific immunity, correct? It is a pathogen specific immunity. So for this particular immunity to develop, unlike our innate immunity, in innate immunity it is like God gifted. Like we have skin, okay, we have cells, and we have some physiological modifications that protect us irrespectively. Whereas when it comes to acquired immunity, this develops not by birth, by the course of the lifetime, the environment I live in, the food that I eat, okay, how much I interact with different environments, okay, the type of water I drink, okay, and the community I live among. All of these keeps on adding up for your exposure to this environment, right? So as you keep on getting exposed to different environments, all right. So now what happens? You are, it's very certain that these pathogens can enter your body either by food, by water, by air, by contact, etc. So the first time, whenever you come across this pathogen, okay, for the first time in your life, we call it as first encounter. Okay, so this first encounter will result, okay, in a type of immune response called as primary response. Okay, when we first get an infection, for the first time when we get an infection, okay, our body produces a response called as primary response. What is primary response? Our body really do not know what it is fighting against. Okay, it do not know how this pathogen will affect our physiology or how it affects our body. So it launches a very low intensity, okay, and a mixed response, okay, very low intensity for a very brief time, okay. So it will launch this low intensity primary response, which lasts for many days, okay. It's very, very slow, all right. Now, it might take a longer time to cure or heal from this particular encounter from this pathogen, but after a few months, after a few years, okay, you get the same infection, okay. Then when you get the same infection for the second or third and the fourth time, the time taken by you, okay, to heal is faster than the prior ones. You might have observed your childhood or the children that are living in your home. They're frequently having cold, okay, they're frequently having stomach upset, they're frequently having some fever or some of the other infections. As they grow up, you find that these infections that recur, okay, recurrence of this infection keep on reducing. Look at yourself. Are you sick every week like children? No, right? So now you have reduced the amount or the intensity or the recurrences of these infections as you grew up. How is it possible? This is possible because of second and third and fourth encounters. Okay, the more encounters that we have with this antigen, our body will build this strong memory about this particular pathogen, So once it builds a very strong memory, okay, now the moment it comes, it launches a very strong secondary response in a high intensity. It is quicker, it is effective, and it will neutralize the pathogen as early as possible. Okay. Now, who carries out this primary and secondary response? So the acquired immunity is basically, basically it is taken care by two important types of cells. Those are called as B lymphocyte cells and then T lymphocyte cells. Okay. So let us study what are B lymphocytes and what are T lymphocytes. Now, these B lymphocytes and T lymphocytes are the type of lymphocytes. All right. So now, when we talk about B lymphocyte cells have the capacity to produce, okay, small proteins called as antibodies. Okay. So these proteins, they can recognize, okay, the antigen by going and attaching to the surface of their cells or flagella or any of those things that they can go and attach to. All right. So the proteins that the antibodies that we talk about, right? So they have the very specific site that they can recognize and they can bind to this antigen. So this acts like a signal to recruit, okay, the other immunologically important cells, okay, and assemble them and neutralize them. All right. So this is about antibodies. Next, when it comes to T lymphocytes, they help B cells to produce antibodies. Actually, T lymphocytes have a lot of functions. They are responsible for giving our body an ability to differentiate between what is our own cells and what is our outsider cells, or if you can tell self and non-self cells. Self means our own cells, non-self means it is from the outside. So this capacity for us to understand the self and non-self comes from T lymphocytes. Okay. Now, as we were talking about, this, acquired, as we were talking about this B lymphocytes, I mentioned that it produces something called as antibodies. So let us see what is an antibody before we understand this type of, this specific immunity. All right. Structure of antibody, okay, contains 4 polypeptide chains. As I mentioned, antibodies are small proteins. Okay. These small proteins contains 4 polypeptide chains, as you can see in this picture. So among these 4 polypeptide chains, there are 2 light chains and there are 2 heavy chains. Okay. Now, the 2 light chains are identical to each other and the 2 heavy chains are identical to each other. Okay. Both the lights are identical. Both the heavy are identical. But if you see in the picture, the light chain and the heavy chain are held together by a disulfide bridge. Okay. Now, there are several types of antibodies that are produced by our B lymphocytes, each having a specific function. For example, we have IgA. Ig stands for immunoglobulin. IgG, IgM, IgE, and IgD are certain types of antibodies. For example, if you talk about human milk, the cholesterol, okay, when we have studied in the chapter human reproduction, the importance of mother's milk in the initial days after delivery, after parturition, that the mother has to give the milk to the baby. Why? Because it is rich in IgA. It gives the ability for the baby to defend itself. Correct? No? So when we talk about allergic reactions, so allergic reactions are the resultant of immunoglobulin E. Okay. Similarly, each of these Ig or immunoglobulins participate in a very specific immune response. All right. So if you see antibody have a particular site in this protein where you have binding regions of antigen. Okay, they can go and bind to the surface of the antigen. So this capacity for them to bind to the surface of the antigen is what sets a response of immunity. Okay, it's fine. When we talk about acquired immune response, so there are principally 2 important types. The first type is called as humoral immunity Humoral immunity, it is also called as antibody mediated response or antibody mediated immunity. Okay, let us repeat this. Under acquired immune responses, we have two types. One among them is humoral immunity, you can call it as, or you can call it as antibody mediated response or antibody mediated immunity or AMI. Now, if you see this, okay, one second. Yeah. If you see this, the name itself has the answer, right? Antibody mediated. Okay. Antibody mediated. So now you can just try to relate who is responsible for producing antibodies and which cells participate in humoral immunity. Correct, isn't it? Right. So next we have the other type that is called as cell mediated immunity, or it is also called as cell mediated response. So popularly known as CMI response. Okay. That is called as cell mediated immune response. So we understood the types of immune responses. Now let us study individually what are these immune acquired immunity types. First, let us talk about humoral or antibody mediated immune response. As I said, the name itself suggests how it initiates. It is initiated by antibodies, correct? No. So what happens? These antibodies, which are smaller proteins produced by B lymphocytes, they are released into the blood plasma. So as they move around in the blood plasma, now these antibodies, okay, whenever they encounter a pathogen such as a virus, okay, so now our antibodies, they have a specific type, okay, specific site, sorry. So these specific sites where they interact with the virus, viral parts called as, wait, let me write it here. Okay, this is your antibody, correct? This is your antibody, and these are some of the regions. Okay, here are some of the regions where your antibody can recognize the virus. All right. So whichever they go and attach, it is called as paratope. Okay. And whatever the site that the antibody has, it is called as epitope. All right. So now these epitopes will go and recognize the paratopes, and then they make a connection. Once they make this connection, now this will drive these cells. Okay, what type of cells? For example, you can talk about neutrophil cells. Okay, so these neutrophil cells, okay, get attracted to these epitope paratope interactions. Now the viral cells are engulfed and they are digested. Okay, so let us talk again. So when we talk about our antibodies can coat or go and bind to specific regions, okay, on the surface of the antigen. It might be anything, okay, it might be polysaccharides, okay, it might be cell surfaces or the cell wall, it might be flagella, anything, okay. This ability for them to recognize and go bind. So once they attach to these recognized surfaces, okay, this we call it as epitope paratope interactions. So once they go and lock each other, they just like lock and key. Now some of the immune cells like neutrophil cells are driven towards this interaction, and then they engulf along with this attached antibodies. Okay, they get engulfed and they are neutralized. So this type of specific response, we call it as humoral immunity. Okay, let us talk about cell mediated immunity. So cell mediated immunity have a very elaborate applications or functions in our body. So here, when we talk about cell mediated immunity, the principal cells which participate in cell mediated immunity are called as T lymphocyte cells. Okay, T lymphocyte cells, what they do is, as I said, they give our body an ability to recognize what is self and non-self cells. Okay, so this also has certain disadvantages and its own advantages. For example, so this see, the level of recognition is you can't even believe how specific they are. Even though we take the organs or tissues, okay, or any of the external, okay, tissues or organs or any parts for transplantation, okay. So for anybody who needs a transplantation, we require a donor, correct? No, so from somewhere we get these tissues or organs. So this is called as donor, correct? No. So whenever these donor organs or tissues are transplanted onto the recipient or the patient who is receiving it, so they undergo this changes called as graft rejection. What is graft rejection? See, our body is very highly specific. A small protein, okay, a small polysaccharides or a different type of lipid or anything, okay, that is not relevant to our body is recognized, and our entire immune system, especially our cell-mediated immune system, goes really against those grafted tissues. Okay, so whenever they go against them, they try to kill these tissues, which leads to something called as graft rejection. So that is why it is so hard for us to find a tissue matching or organ matching a patient, okay, who is going under transplantation. Or even take an example of blood donation, right? For example, if you want to donate blood, or if a patient has to receive the blood, so can you give any blood group to any individual? Definitely not, right? So we look at the blood groups, whether it is A or B or AB or O. Okay, based on this only we will try to infuse the blood into the patient, isn't it? So all these rejections happen because of our cell-mediated immunity, which has a capacity to recognize self and non-self cells. So how do they make it? For example, let us take, already we have a host cell, okay, our own cell, which is infected by the viruses. Okay, now these viruses, they usually produce a specific protein called as MHC proteins. Okay, so these MHC proteins are antigen proteins. Let us say that they are outsider proteins. So they are recognized by your cell-mediated immunity by a specific type of cells called as cytotoxic T cells. So the cytotoxic T cells, what they do is they deploy, okay, they deploy some of the granules, okay, which go and induce the self to which go and induce the cells to self-destruct along with the virus. Okay, it's like asking the cell to commit a suicide so that it can prevent the infection. Okay, so when a cell's function is completely controlled by some other host like virus, it does not know what decisions to make. So our cytotoxic T cells will tell by producing these granules, which go and enter the cells and ask the cell to commit suicide. So by this, the host also dies along with the viruses. Okay, it will digest the entire thing that is present within the cell, preventing the further infection. So this is one of the ways the cell-mediated immunity works. Okay, fine. I will ask you a small question. What would our doctor do, okay, if a patient receives a transplant? Okay, it might be an organ or a tissue. Would he just transplant the organs and leave it? Definitely no, right? So what would he do? Would he wait for the body to respond against these particular cell-mediated immunity? We wait for it to accept or reject. We can't take that risk, right? Organs are very precious, and the health of a patient is also very important, because at that point they'll be very weak. So to support the patient's body, usually the doctors suggest or the doctors prescribe immunosuppressors. Okay, immunosuppressors are taken for a very prolonged period of time. It might go up to 6 months to 1 year, depends upon patient to patient. So these immunosuppressors will reduce the ability of our body to fight against this graft which they have received. Thereby, okay, it will slowly adapt to the patient's body and finally gets accepted. Okay, one way to allow the graft to be accepted is by giving immunosuppressants. So that means what? They have low immunity at that point. That is why these patients are asked to stay as much indoors as possible and as much clean and sanitized as possible. Okay, so this is about cell-mediated immunity. All right, students. So acquired immunity is a specific immunity. We already know this now. So let us talk about what are the two types of acquired immunity. How do we gain this acquired immunity? Okay, so a person can become, okay, and specifically immunized by two ways. One is active way, other one is passive way. Okay, which you call it as active immunity or passive immunity. All right. So what are we talking about? We are talking about acquired immunity. And I told that acquired immunity is something that we gain over a course of lifetime. Correct? No? As we grow up, we get this ability to become immunologically active. Correct? No? So this we gain it, we get this capacity by two ways. How are those? One is active way, other one is passive way. All right. So both in active and passive, we have natural ways and artificial ways. Okay. So let us see what is active immunity and what is passive immunity. Fine. What is active immunity? The antibodies are produced in the host body itself. That means we ourselves have the capacity to produce antibodies. So this is called as active immunity. Remember, active means our body participates in it, right? So you participate in something, you're active. Similarly, to produce an antibody, our body participates, right? So this is called as active immunity. Okay. So how does this happen? Whenever our body or our host is exposed to an antigen, so it can be anything. Okay. It can be anything. It can be a living organism. It can be a dead microbe, or it might be any foreign particles by dust, pollen grain, okay, or any of the lipids or proteins, etc. Okay, any of these substances enter our body. Now, what by the process of primary and secondary responses, our body will produce okay memory cells, and based on these memory cells, they will be able to develop okay a specific antibody and a specific cell-mediated immunity to act against this particular antigen. Now, as I said, for active immunity as well as passive immunity, we have two ways of achieving it. Under active immunity, the natural ways of becoming actively immune is by, for example, it is, yeah, one second. Hold on. Yeah. See, as I was saying, active immunity, we get it by two ways. One is natural ways, other one is yeah, let's continue. So we become naturally active immune by, for example, as I said, when we are exposed to microorganisms from our environment, right? So during our course of lifetime. So as we naturally gets exposed, as we grow up, right? Our body will become, capable enough to defeat these microorganisms by itself. So this is one way of becoming naturally active, correct, right? So when it becomes, when we talk about artificially acquiring active immunity, okay, have you heard about immunization and vaccines? You remember, like while we were younger, okay, when we were babies, forget it. Okay, we don't remember the vaccines that they made us take. Okay, but when we were younger, you remember that we used to wait for the polio vaccines to come to our doorstep, and they would put a couple of drops into our mouth, right? So I would be very desperate to wait for this polio vaccine. For some reason, I like these vaccines. Okay, so what are we doing here? So we are artificially making our body active against a particular disease. How are we doing? For example, if I take polio, okay, polio vaccine as an example, I was surprised when I first learned about it. What are we doing? Our scientists, they take the real polio virus, which has the capacity to cause a disease, okay, and they attenuate it. What is attenuation? Attenuation is a process, okay, in which you make a microorganism inactive to cause, or inactive to to cause an infection. Okay, it cannot cause a particular disease. It can be alive, it can be dead, or it can be a part of an antigen, like it can be a cell wall, it can be a polysaccharide or a lipid or a protein. Any of a part of this particular antigen, okay, is deliberately introduced into our body. For example, COVID. Okay, in COVID also you took some part of COVID virus. Okay, Polio, you're taking polio virus itself. That is called as live attenuated vaccine. That means the virus is alive, but the virus do not have the capacity to cause a disease. You understand my point? So by this, when you are artificially introduced, okay, to the particular pathogen over a course of time, okay. For example, when you are taking COVID vaccines, did you take only one shot? No, right? You took 2 or 3 vaccinations called as booster dose. That means what? You're boosting your immunity by introducing this pathogen periodically, correct? No, as you introduce this pathogen periodically, our body will be able to build a stronger memory cells. Okay, so that is how artificial active immunity works. And example is immunization, or it is also called as vaccination. Okay, all right. So let us talk about passive immunity. Passive immunity means our body is not participating. Okay, so under passive immunity, ready-made antibodies are given to our body directly. All right, ready-made. So do not compare active and passive based on the names. Both are equally important for us to have a healthy life. Let us see here. For example, for a person to gain passive immunity, even we were babies. Okay, while we were babies and we are born, the mother gives us milk. Okay, and this milk we call it as colostrum. Correct? No. So this colostrum, when the baby is so small and so new to this environment, it does not have the capacity to protect itself from various antigens or this environment that it is exposed to. It is very vulnerable to infections, and it is very dangerous for the baby. So how can the baby protect itself? It cannot protect itself. The mother helps the baby to protect. How does it happen? So mother, while she gives the milk, correct, right? See, we are talking about natural ways of gaining passive immunity. Okay. So under that, when mother gives milk to the baby, right? The milk of a mother contains IgG antibodies. Okay. So these IgG antibodies from the mother, okay, it goes through the mother to placenta, and placenta to the breast milk, and to the fetus, basically. Okay, so this IgG antibodies are produced by mother, and through placenta it goes and enters the fetus when it is baby. Whereas when we talk about colostrum has IgA antibodies, and that is given during the feeding time for the babies. So these are the 2 examples for natural passive immunity. One is IgG antibodies, which are produced by mother. It crosses the placental barrier and goes to the fetus. Second one is the colostrum, which contains IgA antibodies, okay, which are given to the infants through the milk. Okay. Next one, artificial passive immunity. Okay, we have 2 examples. One is a tetanus infection. Whenever we have a tetanus infection by fall or by cut of an iron blade, so usually our tissues rot and it will produce a lot of pus. Okay, and it causes fever and chills, etc. In the later stages, it will cause a severe blood poisoning. So to prevent that, ready-made antibodies are given to our body in the form of anti-tetanus serum. It's very, very important examples asked in several exams. Okay, anti-tetanus serum is an example for passive immunity. Okay, remember this. And the second example is anti-venom. Okay, have you heard what happens when people are bitten by snakes? When people are bitten by snakes, they are provided anti-venoms. Okay, specific anti-venom, ready-made antibodies against that particular venom is given to the individual. So that will neutralize this venom and immunize the person, so that he is not affected by the poison. Understood? So this is about passive immunity. All right. So as we studied about active and passive immunity, so let us understand what is immunization. So already I told you immunization is also called as vaccination. What is immunization? Immunization is based on boosting up the memory of the immune system. And how do we do this? One is through active immunization. Active immunization is the example for vaccination. So vaccines are prepared. Okay, vaccines are prepared by isolating an antigen. Okay, antigen. For example, it might be a whole antigen. It might be an entire virus or entire bacteria, or it might be proteins of the pathogens, okay, or proteins of an antigen, or it might be the polysaccharides that are present, anything, okay, that our body can recognize. So these parts are introduced into our body, okay, through vaccine process. So once we have taken this, now our body will go under primary response, okay, and booster doses will give us secondary responses. So as our body is being introduced to this pathogen, now our body will be able to generate two important types of cells. One is memory cells, okay, memory B cells, and the other one is called as T cells. So these memory B cells and the T cells that are produced, okay, by our body, they will give our body the ability in the later stages, if you are infected by a real antigen which has a capacity to cause an infection, okay, so they will be able to recognize this pathogen as quickly as possible and neutralize them as early as possible, and protect your body from these deadly diseases, okay. So this is about active immunization. One is vaccination. Example is polio vaccine, hepatitis B vaccines, and DPT vaccines, correct? No. So one way of today, we are using recombinant DNA technology to produce vaccines. And one of the examples of the vaccines that are produced by DNA recombinant technology is hepatitis B vaccine, okay, which is produced from yeast. So remember this example. What type of vaccine is developed through recombinant DNA technology? So you have to tell it is hepatitis B vaccine, okay, which is produced from yeast. Very, very important. Okay. So we talked about active immunization, correct? No. When we talk about passive immunization, it's simple. Preformed antibodies, for example, antivenom, okay, is injected into your body, correct? No. So this is called as passive immunization that can save life in a very quick way. Okay. So for passive immunization, your example should be antivenom for snake bites and tetanus, okay, anti-tetanus serum for tetanus infection, okay. For active immunization, we inject a part of antigen or a whole of antigen, okay, and introduce our body and make our body build its own antibodies. When it comes to passive immunization, already preformed antibodies, okay, are given to the person, okay, so that they can have the quicker immune response. An example for passive immunization are tetanus, snake venom, etc. All right. Okay, students. So we have to talk about allergies. So we understood how our immune system helps our body to protect itself, to become so specific against something. Okay. And from the birth, okay, before the birth and all the way we lead our life, immune system keeps doing this job of protection, correct? No. So we think the immune system, we actually owe a great deal to our immune system for protecting our body all the time, 24/7. But what happens if our immune system do not function properly? Or what happens if our immune system overreacts to a certain situation? One of the examples of immune system impairments, we can call it as, is allergy. Allergic reaction is, in simple terms, if you tell allergy, it is called as overacting or overreaction. So what happens in allergy? Exaggerated immune response, we call it as. What is exaggeration? For example, it might not be a very serious problem, but you exaggerate thinking that, oh, it's a very big problem, and you panic. So when your immune system panics, this is called as exaggerated immune response. So whenever our immune system exaggerates for some antigens, so it might be dust particles. Sometimes you might have had outside food. Okay. You might get severe rashes on your skin, or it might be the dust from the street or the pollen grains that have entered in your nasal cavity. All these small amount of antigens that are in our environment that enters our body will trigger an exaggerated immune response, and that will cause certain symptoms of an allergy. Understood? So anything that causes these allergic symptoms are called as allergens. All right. So under allergy, we have allergens. We have mitts in the dust, pollen grains, animal dander, fur, okay, etc. Now, what happens in allergy case? in allergy, our body our antibodies, a specific type of an antibody is produced in a very high amount. What type of antibody is produced in a very high amount? IgE type of antibody. Remember this. IgE antibody is responsible for causing an allergic reaction. Okay, antigen comes. So our B lymphocytes think, okay, it's a very big infection. So it will produce a lot of antibodies. What type of antibodies it produces? IgE type of antibodies it produces. Once this IgE type of antibodies are produced, they go and bind to this specific type of cells called as mast cells. They go and bind to the mast cells and ask the mast cells to release a specific chemical called as histamine. Okay, and it also produces serotonin. When it produces histamine and serotonin, which are quite acidic when they increase in the blood, that will cause allergic reaction. Understood? So that will cause what? Some of the symptoms like sneezing, watery eyes, running nose, difficulty in breathing, wheezing, skin rashes are all the examples. Okay, have you seen people become asthmatic? Okay, the people with asthma problem, they also that they also suffer because of allergic reactions. Understood? Sorry. Fine. Okay, we have, yeah. Now, how do we know that a person has an allergy? So a small doses, for example, if you go to hospitals, the doctors will, before giving you any medications like such as antibodies or anything, antibiotics or anything, what they will do is they will inject you with very, very small doses of this antigen onto your, on your skin, and they will circle them. Okay, they will circle it and write the name of this antigen. All right. So as they write the name of this antigen, they will leave for some time. And if you develop any rashes, okay, or blisters or itching or burning sensation in that injected area, then that means you are very sensitive to that particular antigen or allergen. Understood? So that is how we know if a patient is allergic to certain foods or medication or anything. Understood? So how do we treat these allergies? Whenever, even if you had any allergy, if you had gone to medical shop or the doctors would give you something called as antihistamine. Correct? No? We understood mast cells will produce histamines. So one way to reduce the allergic reaction is by giving the patient antihistamines. Understood? And they are also given adrenaline, okay, steroids. So they quickly reduce the symptoms of allergy. Understood? As I was saying, for allergies, asthma is one of the bigger problems. So asthma is a respiratory disorder that is due to the allergies, which is causing inflammation in the lungs or swelling in the lungs. You might have seen some of your friends being allergic, okay, and they might have asthma situations, or you might have seen some of the people having this asthma around you. So that is due to their environment that they're grown in. Usually we have very less interaction with our environment. We don't go out, we don't play, we don't get our hands dirty, we don't sweat, okay, we don't eat properly. So when we are not exposed to more environmental diversity, our body, it becomes more and more sensitive, okay, to the environment. As our body becomes more and more sensitive to our environment, it will not accept even small changes in the environment. So that will cause a serious problem in having a very healthy life in our later lives. Understood? So this is some of the aspects of allergy. Okay. And the second example for your immune system disorder is autoimmunity. Okay. Now you remember when I talked about your body's ability to recognize self and non-self cells, right? So you might have neglected this aspect. Okay, Leaf, what is there to recognize who am I and who is somebody else? What if the system fails? What if you lose the ability to recognize who are your friends and who are your enemies? Right? With the panic, you might attack your friends also, or with the acceptance, you might accept your enemies also. Two ways possible. Correct? No? Similarly, our body will lose the ability, okay, to differentiate between what is our self cell and what is our non-self cells. Okay, let us see what is happening here. So in the memory-based acquired immunity, our ability to recognize the self cells from foreign cells is lost. There are so many different, reasons for how a person will become autoimmune, okay, to a particular disease. For example, you have many examples like psoriasis is one of the examples of autoimmune disease, rheumatoid arthritis, okay, Alzheimer's is another example for autoimmune disorders. So many reasons are there, and most of the reasons are due to genetic reasons, okay, and unknown factors, okay. This causes our body cells to attack their own cells, okay. It might be the cells of brain, it might be the cells that is between the bones, okay, or it might be the epidermal layers of your skin. All these attacking our own cells is called as autoimmunity, okay. How can we define autoimmune disease? The ability of our body, okay, to differentiate self and non-self cells is lost. So, okay, therefore, our body cells will attack our self cells, resulting in the damage to our body. And this is called as what? Autoimmune diseases. An example is rheumatoid arthritis. Okay, so what is happening? The junctions between the bones, okay, are corroded because our cells, okay, osteoblasts are attacked by our, by our immune cells. So that will lead to bone degeneration. And a person who is suffering from rheumatoid arthritis have low mobility. They have excruciating pain for normal movement, walking, folding, eating. They can't fold any of the joints or move any of the joints, and they have excruciating pain. So that is autoimmune disorders. Okay. So you have the question now. Now it's time for quiz round. Okay. So you have the first question. The type of antibodies produced during the allergic reaction. So it is a very simple question we discuss right now. Let us see how many of you answer. Okay. Yes, your polls are live. You have 1 minute to answer this question. Go on. Let's see how fast you will answer. It's a very direct question. The type of antibodies produced during the allergic reaction are, okay. Make it fast, students, okay. It's very easy. So I'll end the poll. Your time is up. 1 minute. Okay. So if you have chosen IgE as your option, okay, that is the right. Okay. Option D is right. Correct. Now what is happening with respect to IgE? IgE antibodies that are produced, they go and bind to the mast cells. Mast cells will produce histamines and serotonin. So that will lead to allergic reactions. So, okay, antibodies that are responsible for immune response such as allergies are IgE antibodies. All right, fine. So our next topic is AIDS. We will have an introduction about this topic, okay, and we will continue this chapter or these topics like AIDS and cancer in the coming up sessions. All right, so let us see. Now, when we talk about AIDS, you might have a mixed opinion about it. One thing is it is a sexually transmitted disease. Second thing, it is a deadly disease. Some people might think AIDS is very, very dangerous. So we should keep these people as far as possible to have this AIDS. Okay. And some people might be not understanding how severe or how serious is this disease. All right. So the knowledge about this particular sexually transmitted disease, okay, AIDS is very, very essential in so many aspects, okay, such as in having a social balance where we treat the patients with respect and dignity. Second one is having a knowledge about this disease can help you prevent any further, okay, ways in which you might be a victim for AIDS. Okay. And the third, education about this will improvise our social response as well as our personal ways to lead a healthy life. Understood? So let us see what is the story behind AIDS. So AIDS stands for acquired immunodeficiency syndrome. Okay, it is a deficiency of immune system. Okay, we know that immune system helps us in preventing any of the antigens that is entering in our body to cause a disease. So if we lack this capacity, we have deficiency of immune system. So AIDS is an example for deficiency of immune system. Understood? Now, when you don't have an immune system or a very low immune system, what can happen to your body? Your body can be victim of any different types of infections, such as viral infections can happen, fungal infection can happen, protozoan infection can happen, bacterial infection can happen. Usually what happens is, if I am having a bacterial infection, okay, my body is already under high alert and it produces a lot of white blood cells to protect my body, right? So it does, that is why when I have one certain type of infection, second or third type of infection do not occur simultaneously. You will not see a patient having malaria and dengue together, correct? No? Or you might not see a person having typhoid and malaria together, correct? Right? Have you ever thought about this? 2 or 3 diseases will not occur at the same time, correct? No? So if a patient shows more than one disease, symptoms of more than one disease, correct, right? So he is showing bacterial infections also, he is showing viral infections also. You are not able to define what type of symptoms these are. COVID, you remember, COVID also we were showing many symptoms, many different types of symptoms. So this we call it as syndrome. Okay, a multi-symptomatic condition is called as syndrome. That kind of state happens in an individual who has AIDS, okay, because of immunodeficiency. Understood? And the causative agent for AIDS is human immunodeficiency virus. Okay, in short, we call it as HIV virus. HIV virus is a class of retrovirus. Okay, so retroviruses will have RNA as genome. Okay, so there is a very interesting story on how AIDS started first, but it was first reported in 1981 in America. Okay, 1981 to 2023 now. Okay, in the earlier census was 2022. So by then, AIDS in 25 years has claimed over 25 million people's lives. Okay, that is why it is such a deadly disease. Now, if I tell you how AIDS spreads, you would be very, very astonished. How is it even possible that this disease, having the having the ways to spread only by a fixed means, could claim 25 million people's life? All right. So let us keep this discussion alive for the next session. All right, students. Thank you very much. I'll see you in the next class. If you have any doubts, you can post your doubts in the chat box, or you can raise your hands. We can discuss about it. All right. Thank you very much.